Good evening, everyone, and thank you so much for joining us here tonight to mark World Cancer Day 2025 here at UCL with our panel event this evening, Transforming Cancer Care, UCL Research Innovations and Discoveries. My name is Jane Ganeshalingam, and I'm an MBPhD student at UCL. That is, I'm a medical student here, and I interrupted medical school to do a PhD at the UCL Cancer Institute, where I researched the development of a type of childhood leukemia using stem cells and genetic engineering techniques. And I'm also your chair for tonight's event. Now, during this panel event, our UCL experts will talk about some of the transformational work taking place in their respective fields and how new and different approaches to cancer will change the future of cancer care. Now, without further ado, I am pleased to introduce our panelists. Firstly, we have Paul Maciocia. Paul is a consultant hematologist and a clinician scientist specializing in chimeric antigen receptor T-cell therapy for cancer. He now sees patients in the CAR-T clinic at UCLH while running a lab group based at the UCL Cancer Institute. Simona Parrinello. Simona is Professor of Neuro-oncology at UCL, and her research focuses on adult neurogenesis and brain cancer. She also leads the Samantha Dickinson Brain Cancer Unit and co-leads the CR UK Brain Tumour Centre of Excellence. Kate Ricketts. Kate is Associate Professor of Cancer Physics at UCL, and she is an NHS clinically accredited radiotherapy physicist. She works at the interface of radiation physics, AI, and cancer biology towards improving patient response to radiotherapy treatment. And last but not least, Chris van Tulleken. Chris is Associate Professor at UCL and an infectious diseases clinician. His research focuses on how corporations affect human health, especially in the context of nutrition. He is also one of the BBC's leading science broadcasters on television and radio, and recently delivered the Royal Institution's Christmas lectures. They're fantastic, by the way, and available on BBC iPlayer if you've missed them. Thank you to our panelists for joining us today. We will now hand over to our panel for their opening remarks so you can find out a bit more about their respective fields. Now, following these remarks, we'll have a discussion amongst the panelists before moving on to the Q&A section with all of you. So firstly, I would like to ask our panelists to respond to the following question. Can you tell us about your research specialism and what developments you have seen that are changing how we approach cancer? Over to you, Paul.
Thanks very much, Jane. So I work in CAR T cells. So I'm a hematologist, a blood cancer doctor, and I also work in the lab. And what we use is a different way of treating cancer. So if you look at kind of traditional ways that cancers have been treated over the last 100 years, there are really three ways. Surgery, cutting it out, radiation, which is where we hit the cancer with radiation, or chemotherapy drugs, the sort of things that we know about. And the way that all of these work really is they work by kind of damaging those cells. So either cutting them out or directly damaging to them. And they're not specific therapies against that cancer. There are therapies which work by damaging those cells and trying to damage them less than you damage, trying to damage them more than you damage the normal cells. And that's been the paradigm for a long time. But what we try to do is something a bit different, which is to use the immune system to kill cancer. So your immune system is what you have to protect you predominantly from infections. And so there's lots of different parts to that. But the main thing that it's trying to do is identify things which are foreign in your body and kill them. You have these cells called T cells and that live in your blood. And what T cells mainly do is defend you against viruses. So you patrol around the organs of the body looking for cells which are infected with viruses and try to kill them. And they're not so good at killing cancer cells because cancer, of course, is just your own cells that have gone wrong. So it's not something foreign. They don't look like something foreign to the immune system. So what we try to do is kind of re-educate the immune system to do that. And this has been a big field recently. So there's lots of different ways that people have tried to use the immune system to kill cancer. So using kind of drugs to take the brakes off the T cells and to make them work a bit better. Using kind of antibody drugs to try and paint cells that can be recognized by the immune system and killed. What we do is even another step different from that, I guess. So what we do is we take the immune cells out from the patient. So we take the T cells out from the patient and they sort of go in a machine that collects a little bit of their blood and we isolate these immune cells and we take them to the laboratory. And what we do in the laboratory is to genetically engineer those cells. So we give them a new bit of DNA code, which makes them express this protein called a chimeric antigen receptor, or CAR. And that's a new set of instructions which tells them to recognize the cancer cells. So that goes into those T cells, and we then go and inject the T cells back into the patient. And once they get there, they're able to go and find the cancer cells, recognize the cancer cells as being something that is now foreign to them, and something that they want to kill. So they kill the cancer cells and they start making copies of themselves. We start off with injecting a relatively small number of T cells and they kind of expand. So it's like a living drug patients, and they can go throughout the body and find where the cells are and kill them. And they can last for very many years. So it's kind of a cool concept, but I guess, does it work? And in some blood cancers, at least, it's had kind of amazing results. So if you look at what normally happens when you first try a new cancer therapy, it's usually in people who have got really very advanced cancers with very few options. And usually it maybe makes a small bit of a difference to what happens. So when CAR T cells were first tried in some people, particularly children with advanced leukemias, took these children who didn't have any real options left, and about 90% of them, their cancer was kind of went into remission. Maybe half of them, it stayed in there long term, so kind of cured. And across different blood cancers, in adults and as well and in children, we're using this regularly in the NHS. What we're doing now in the laboratory to try and make better types of these treatments. So I work on trying to make CAR T cells for other kinds of blood cancers and also some other kinds of what we call solid tumors, so things that don't arise from blood cells, like ovarian cancer, lung cancer are two different areas that we work on in my group. I also work on trying to make CAR T cells from one donor that can treat a lot of people. At the minute, we have to make them bespoke for each individual patient, which takes time, it's quite expensive, and the T cells don't always work well. So it's very difficult, but we're trying to make them from a universal donor. And we're always trying to think of ways to make these therapies safer and more effective and be able to treat more patients. So that's what I'm doing, and that's what I hope is going to be a big part of the future for cancer therapy at UCL and throughout the world.
Thank you, Paul, and thank you for that great introduction to CAR T cells. Simona, over to you. Can you tell us a little bit more about your research specialism?
Yeah, of course. Hello, everyone. So I am a biologist specializing in stem cells and brain cancer. And I'm a basic scientist. My lab is based at the UCL Cancer Institute. And the main focus of our work is on a brain tumor called glioblastoma, which is the most common and aggressive type of brain cancer. And it is really, which means it's a type of cancer that forms within the brain. And glioblastoma is a really devastating disease. It's one of the most aggressive forms of cancers in patients. And it is a prognosis that has remained unchanged for many decades, a pretty poor one. On average, patients survive about 18 months post-diagnosis. In addition, it's a disease that robs patients of their identity and independence because it affects the brains and it comes with a series of cognitive impairments. So it's a really terrible disease, and a disease in which we need to do a lot better through research. And I think that one of the key problems here is that by the time the tumors are typically detected in patients and treated, they're very advanced. And as such, the tumor cells have developed a series of mechanisms that make them really resistant to the current treatments that are available, some of the ones that Paul was referring to, such as surgery, chemotherapy, and radiation. And so it's really a point in the disease stage where achieving a significant improvement is extremely difficult. And so, of course, an obvious way around it would be to try and treat disease much earlier. But that comes with a series of challenges as well. The biggest one is that we know quite a bit about the late stage disease. This is the stage of the disease at which most of the cancer research, and definitely in glioblastoma specifically, is focused. But we know a lot less about earlier disease stages. And this has largely come from the fact that we don't have access to patient material at these early disease stages because it's simply not taken out. And we also have not had availability of very powerful preclinical models that mimic the disease well enough so that we can study those for understanding its biology. But this has begun to change, and work from my lab and others has developed much better models of the disease. And we have begun to study these. And these have unlocked the biology of these earlier disease stages. And as we've begun to do this, we made some really exciting discoveries. Particularly, we are beginning to realize that these early stages of disease have a lot in common with the way in which the brain responds to an injury, and also a lot in common with many neurodegenerative disorders. And the more we understand about this and the programs linked to neuroinflammation, the more this reveals ways in which we can target this, and therefore identify and target the disease at a much earlier stage, where we can have a much greater impact. And so this is really what we are focusing on at the moment. We think that these discoveries are a major breakthrough and a way in which we can finally begin to crack this very important problem. And but the more we understand these programs, the more we will identify ways in which we can, for example, develop screening tools for patients that can identify the cancer much earlier, before it has the chance to progress to a very advanced and intractable disease stage. And we can also identify new treatments that can eradicate these early cancer cells.
Thank you very much, Simona. Your research sounds very exciting. Kate, could you tell us a little bit more about your research?
Oh, yeah, with pleasure. Great to be here this evening, by the way. So my research field is radiotherapy. So this is where we use radiation, a scary word to some, an exciting word for me. So we use radiation, usually X-rays, but we are lucky enough to also have protons to kill cancer cells. So half of all cancer patients will receive radiotherapy as part of their treatment. So, I think it's very important to get it working optimally. There have been huge technology developments over the past 20 years in radiotherapy departments across the world. In a radiotherapy department, you will find the most high-tech and complex machines that you could find in a hospital. They use particle accelerators to accelerate electrons and protons to near the speed of light. So for physics geeks in the audience, so we're looking at relativistic, so Einstein level physics. And they're using the same tech that are used by particle physicists, at famous centers like CERN. But all of this is under Tottenham Court Road, where we work. Our proton therapy gantry is 4 stories high. It weighs 100 tons. And so this 100 tons of steel are rotating around the patient to deliver a proton beam with millimeter precision. So this is serious engineering challenges that have happened to achieve this. We've seen huge computational developments that have really revolutionized what we can achieve in radiotherapy. So we've now got algorithms that can optimize the dose delivery to the patient. So we can use 3D images of every patient to build a 3D model of the unique anatomy to that patient, to tailor where to maximize the dose to the tumor and where to minimize the dose to everywhere else, so you can minimize side effects. So what we've got is a very fine paintbrush, if you like, that we can paint any dose distribution onto the tumor. Now we've got that fine paintbrush, the question is, where do we put the dose for optimal outcomes? So what we're doing now is we're needing to integrate all of the latest cancer biology and clinical knowledge into guiding where to put this dose. So we currently personalize dose delivery to these 3D models of anatomy, but anatomy is quite a physical thing. We want to go one step further now. It's quite a big step. And personalizing the dose plan based not just on anatomy, but also on the tumor's unique biology. So cells can have different gene expressions, and they're also in different environmental conditions, like oxygen levels, and all of these impact how sensitive or resistant the cell is to radiation damage. So we're now building models to predict these differences in response so that we can inform treatment decisions of the radiotherapy doctors, to identify early markers of treatment response to give us a chance to change, to adapt treatment during the course of radiotherapy. So how are we actually approaching this? It's all about the unique and rich data that's produced during a patient's journey in a hospital. So they come in for diagnosis, they get images, they'll get a biopsy, maybe some genomics for some cancer indications. There's lots of clinical notes generated for all appointments. We've also got digital dose maps in radiotherapy, and all of these together are formed for each patient to form what we think of as a unique digital biobank of data across multiple scales. So the cell scale, the genomic scale, the tissue scale, and also multiple time points. These data sets are really diverse. So we've got images, we've got text, we've got long lists of numbers for genomics, and they are big. So we can't really sit down with pen and paper to organize these. A perfect tool to handle this data is AI. And so we're using machine learning to try and integrate and analyze this huge level of complexity. So at the moment, we're working on data pipelines and AI tools so that we can inform where to paint our dose, and that we can finally bridge that big gap that currently exists between these big physics and engineering technology advancements in radiotherapy and the latest cancer biology knowledge and outcomes data that's at our fingertips.
Thank you, Kate. That sounds very amazing and very exciting indeed. I can't wait to hear more about it in our discussion coming up. And Chris, could you tell us a little bit more about your work and any developments you have seen that are changing how we might approach cancer?
Hi, everyone. It's lovely to be involved in this. So I'm an infectious diseases doctor at UCLH, but my academic work at UCL focuses on global health and particularly food and nutrition. And that may sound incongruous, but of course, nutrition affects your risk of infection. And working in low income settings early on in my career, I saw that it was diet particularly that affected mortality in infants. And this was because of the marketing of baby food to populations that couldn't afford the food, didn't have clean water to make it up, couldn't often read the instructions about how to make it up. And so my research morphed from being about viruses and molecular virology into being about what we call the commercial determinants of health, how big corporations affect our health in good ways and in bad ways. And this is part of one of the many risk factors that inform our risk of dying of lots of things, but particularly cancer. So I study nutrition and food at a macro scale. I study the food system, but also the peculiarities and particularities of the food we eat and how we define unhealthy food and unhealthy diets. So in particular, I've done a lot of work on a relatively new way of thinking about unhealthy diets, ultra-processed food. And there is no perfect way of defining either an unhealthy product nor an unhealthy dietary pattern. But we now probably have more evidence around an ultra-processed dietary pattern being harmful to human health than we have around any other dietary pattern. When we talk about the Mediterranean diet or ketogenic diets, low carb, high carb diets, Western diets, junk food diets, we have more formal evidence around UPF than anything else. And it's a developing field, but there are a lot of studies, mainly epidemiological, but also some experimental studies that are linking increased consumption of ultra-processed food with different kinds of cancer, but particularly gastrointestinal cancers. Now, ultra-processed food is broadly defined as we can think of it as industrially processed food. It's food, the definition really means food with additives that you don't find in a domestic kitchen. And it's a way of describing a modern American diet. So it's a very large range of products. And the evidence all applies to that dietary pattern. In the UK, around 60% of our calories on average come from ultra-processed food. And the same is true in the US and Canada and Australia and lots of the global north. And we see in some cancers a dose response as people eat more ultra-processed food, so their risk of some cancers goes up. And that's particularly true of these gastrointestinal cancers. And it's concerning because over the period that we've been eating more and more ultra-processed food, we've seen, and again, this is a developing field, evidence mounting that rates of young onset cancer incidence are increasing, particularly in the same countries where lots of UPF is eaten, and particularly in young people who eat more UPF and have eaten more UPF as a proportion of their body weight than older people. And we see this particularly with the gastrointestinal cancers. So we have some epidemiological evidence from quite good prospective epidemiological studies, and we also have more and more plausible mechanisms when we start to worry about how a risk or a stimulus might drive cancer. We have to think very carefully about whether it's truly causal or whether it might just be associated. Do cigarettes cause lung cancer, or could it be the matches that cause lung cancer? And disentangling those problems is complicated, but that's what epidemiologists are really good at. And part of trying to figure out causality is saying, is there a mechanism by which the food might do this? And we actually now have a growing number of mechanisms. So we know that ultra-processed diets or unhealthy diets are inflammatory, and that's linked to risk of cancer. We also know some ingredients in the food are carcinogens, and some of the packaging, the molecules that leach from the plastic packaging, particularly after microwave cooking, they may be carcinogenic. Some of the sweeteners have been shown to be carcinogenic. We also know that obesity itself is a risk factor for cancer, as an independent risk factor. And this, more than anything else, is food that is engineered to drive excessive consumption. So we have a lot of growing evidence around how diet links to cancer. It's all relatively early, but I think at the moment it warrants a precautionary approach. And the study, the type of studies that I do around the food system and my colleagues that I work with, what I think the ultra-processed food science reveals really is that many of the ill effects of ultra-processed food, whether we're talking about weight gain or metabolic disease, cardiovascular disease, or cancers, we should best think of them as commerciogenic. And this was a really important turning point in the pandemic of tobacco-related lung cancer, was when we started to frame it not as a problem of individual responsibility, where smokers had chosen to smoke and therefore it was to some extent their fault that they got lung cancer. There was, by the 90s and early 2000s, a really widespread understanding that cigarette smoking is driven by an industry that profits from marketing and selling cigarettes. And we see the same thing in the food system, that the food system is extremely financialized. And so that tells us if we want to reduce risk of cancer, the kind of policy interventions we need to look at, and we need to stop thinking of diet-related diseases, including some cancers, as being issues of willpower or responsibility, and things that much more need policy solutions to change the structure of the food environment that controls what people eat.
Thank you for that, Chris. It's very interesting to hear about the global health and policy perspective on cancer research. So thank you all for your fantastic opening remarks. And before we move on to the audience Q&A session, I would like to ask our panelists a couple of questions. So firstly, what role does collaboration, be it between individuals, disciplines, or institutions, play in accelerating innovation and discovery in the field of cancer research? Paul, would you like to start us off?
Yeah, I mean, I think you realize if you try and work in a silo, and you're yourself, you probably aren't going to get the best out of things, because it's not possible. And it's not possible to be excellent at everything, and there will always be things which other people will either have skills that you definitely don't have, or a fresh viewpoint. So, we collaborate within UCL, hopefully sort of nationally and internationally. And sometimes you make very surprising collaborations that you wouldn't have sort of thought of. And then you meet someone at a meeting or something, and actually you didn't think your two fields were linked at all, but actually there's some synergies there. So that's my take.
Thank you. Kate, you mentioned your introduction about your research sort of beginning to link cancer biology and radiotherapy and things. Do you have anything to add with respect to the importance of collaboration in between institutions and disciplines and accelerating?
Sure. Sure. I mean, it's evident that for radiotherapy you need collaboration. So there's engineers, nuclear physicists, particle physicists, and then the clinical side, cancer biology. It's all together. But now, I mean, the research I'm looking at, if we didn't have collaboration, there would actually be a major roadblock to me doing my research, because machine learning models, they're very hungry for data, and one institute, one department can't produce enough data. You need lots of data because the machines need to look at and recognize lots of repetitions of patterns. So you need to go through the patterns lots and lots of times. But with a small data set, you can't do that. So you can't actually teach the machine to learn and recognize patterns. And also you need to actually make it fair and representative of the populations we're looking at. So if you've got small data sets, you're going to be leaving out some underrepresented patient groups. So you need to make sure you're training these models so that they're non-biased. For example, ethnic minorities, if you've got small data sets, you're not necessarily going to capture those. So it's really important to get big data sets, collaboration. So we need to bring institutes together to kind of share these data sets and techniques to do that. It's not easy.
Thank you. And Chris, do you have anything to add from a global health policy making sort of perspective?
No, I love just sitting in my silo. I don't want to talk to anyone. No, I mean, obviously, I'm someone who moves, my clinical discipline is quite relatively weakly linked to my academic discipline. And I want to make the case, I suppose, if we want to link diet and cancer, we need to understand the agricultural practices used to produce our food, the pesticides and agrochemical inputs that go into production, all the way through to the packaging molecules, through to the way people cook it, and then many of the putative mechanisms by which poor diet drives cancer are quite indirect. So there are some things that you consume where you could point it and go, that might be directly causing cancer. But let's say we have a food or an additive or a dietary pattern that drives thinning of mucus in the colon or colonic leakage of bacteria. We could see that you get it's not that the food itself is causing the cancer; it's that the movement of bacteria from gut to liver drives a cycle of inflammation that then causes cancer much more indirectly. Or we might see that the links to obesity or inflammation. Similarly, we might see that. So really understanding with the microbiologists, the gastroenterologists, the gut physiologists, and the cancer biologists, all of them are going to have a massive impact on how we start to consider the plausible risks by which food might drive cancer.
Thank you. And Simona, from a cancer biologist point of view, do you have anything to add on how collaboration might accelerate your cancer research?
Yeah, I mean, I think I agree with everything that's been said. It's fundamental to the way we do research now. And I think a lot of it comes from the staggering advancement in technology that we've experienced in biology over the last 10 years or so. And the techniques become more and more sophisticated, but also more and more complex. And it's impossible for a single lab to be able to do them all. And so by necessity, you will have to collaborate with experts in the field to also advance science much faster. But I think also in the context of cancer research is also really key to sometimes team up between more basic biologists and clinicians. I think I have a really good example of that within my team, where we work incredibly closely with a neurosurgeon who's also a clinician scientist. And ever since we joined forces, his name is Kieran Hill, and it's been an amazing success of being able to work closely together. We bring the biology perspective, he brings the clinical perspective, the in-depth knowledge of what the real clinical issues are for patients, and I think it works really well for cancer research in particular.
Thank you for that, and thank you to all of our panelists for providing plenty of food for thought. And now, as we look to the future, what advancements, priorities, or considerations do you think will transform the management of cancer over the next decade? Chris, would you like to kick us off?
Can I ask someone else to kick us off? Because I actually do you mind me doing that, Jane? Is that a cheeky thing to do? In a way, I kind of want to hear other people's answer to this question, if that doesn't seem like I'm dodging it. I am dodging it a bit, but I'd rather reflect on someone else.
No, of course. Okay. Kate, would you like to
Yeah.
start us off?
I shall kick us off.
Yeah.
So, there's huge. You could go on for hours in answer to this question, but to pick a couple, looking at all of this data, this multi-scale data that we're looking at, including, there's a real underuse of patient-reported outcomes in deciding treatments and in adapting treatments during. And I think, as survivorship continues to improve, cancer treatment is getting better. We're getting more and more successful. Survivorship is improving. And I think these patient-reported outcomes are really going to grow in importance. We're not only looking to treat the cancer, we're looking to, consistently give a good quality of life to the cancer survivor. And ways of doing that and ways of integrating that into treatment decisions, I think, is going to be quite important. Something we're looking at with our data pipelines as well.
Thank you. Paul, do you have anything to add?
Yep, sure. I'll take on this challenge, Chris. Thank you. So I think one theme for cancer therapy as a general is much moving towards what we call personalized medicine, and that's a little bit of a buzzword in cancer. But what it means really is that if you look at even how we define cancers, a lot of them are defined a long time ago by what they look like under a microscope, and now we know that actually genetics of different cancers, they even don't look the same, are totally different. They're actually different diseases that look the same. So cancer is like it's a million things. There's a lot of different things. And so moving towards therapies which either use drugs which target particular pathways, or to plug my own kind of area, that use the immune system to do it in at least a bit more of a targeted way, I hope is going to be much more part of the future, because hopefully you're doing that in a more specific way, so you might have less of the side effects. You might have something which is more likely to work for that particular person. And that really fits into this whole big data sets as well that we're accumulating, and we're inching towards that.
I should say I was very pleased. I've saved my answer, and Simona, do interrupt me if you wanna go. Of course, I don't. I do treat patients with cancer, but what I treat is the infectious complications of cancer therapy. And so, from my perspective, either we're trying to give them live immunizations, for example, we're trying to reconstitute their immune system, or we're dealing with infections that cancer patients get that other people don't get. And so, the more we can do the kind of things that Kate and Paul are talking about, hopefully the fewer side effects we're going to see of the cancer treatment. What we really want to do is to kill the cancers, not the immune system very often, and have treatments that don't cause these off-target.
Sorry, Chris, we lost you for a second there.
Am I back?
You are back. Hello.
I'm so sorry. I need to call my network provider. I don't know where I got to, but that's the struggle that my patients are living with is the infectious complications. And so from my perspective, I would love to see both what Kate and Paul both talked about. I think hopefully I would see fewer cancer patients in future.
Amazing, thank you. And Simona, do you have anything to add?
Yeah, I think the future really lies in the context of prevention and this emerging new field that is called cancer interception, which is prevention in a much broader sense, not just about, don't smoke or don't eat processed food, but it's also about trying to capture these tumors, before they really have a chance to transform into malignant lesions. And that, I think, is where cancer research is going as a field, and it's a challenge. But if we can get there, that would really be part of a sort of like, you can imagine a future where, as a routine check, you will get screened for potential early lesions of different cancer types, and then you will be given a drug that kind of stops its cancer in their tracks before they get a chance to do damage and really reach that sort of late malignant disease stage where treatments are much more challenging and unlikely to really cure it.
Thank you. And thank you to our panelists for answering those questions. I'm now going to open the floor to some questions from our audience. So just to kick us off, I have a question for Paul and/or Simona. So how far away do you think we are from delivering CAR T-like therapies and using them in the treatment of other cancers, for example, brain tumors?
Do you want to go first, or shall I go first?
You go first.
Okay, so I think CAR T at the minute is used in the NHS and in other settings in lymphomas and leukemias. And much less, there's no sort of approved products that we would use in any of these, what we call solid tumors, including brain tumors. That doesn't mean that there's not a huge amount of research in this area. In fact, Simona and along with other colleagues at UCL are going to be involved in a CAR T trial of for glioblastoma, which is going to be starting at UCLH this year. There have been some success in this kind of an area in brain tumors, not in the kind of dramatic responses that we've seen in blood cancers, but with responses and patients getting into remission. And that's also been the case with some other tumors. It's a really hard challenge. There's a whole load of reasons why your immune system doesn't want to go to those places. You've got to find molecules which you can kill on cancer cells, which aren't on the normal cells, which is difficult for solid tumors. You've got to make sure the T cells get to places that they don't necessarily normally want to go. They like to go where blood cells are. They don't necessarily like to go to solid tumors. It's a big challenge. I mean, I'm an optimist in this field, so I think it's the future. I think that's what's going to cure all these cancers in the future. But, some people might not.
Maybe just to add, I think the biggest problem for brain cancer, particularly the most malignant types, is that it's a very complicated disease, and the cancer is made up of cells that are all different from each other, and they also change during the course of the disease, and they evolve from one state to another. And so CAR T technology relies on us having a target against which to direct the T cell, and that's very hard to achieve in a complex tumor like glioblastoma, for example, because even if we found a specific target, it's likely that it wouldn't be present on all the tumor cells, and that invariably there will be a population that escapes the CAR T treatment. And so, of course, you can imagine going in with more complex CAR T treatments where you would have multiple cells with multiple targets, but finding these targets is quite difficult. It doesn't mean we're not trying. We're trying very hard. And in combination, in addition to that, glioblastoma in particular is what we call an immune cold tumor, meaning that it has developed a lot of different mechanisms that make it invisible to the immune system. So you have that barrier to overcome as well.
Thank you for that. And I have another interesting question for Simona and perhaps Chris as well from our chat. If one merged nutritional and nutritional data and that pertaining to the diet and the microanatomical data of glioblastoma, for example, could that give a clearer idea for patient treatments? I ask because some diets alter neurological functions. For example, the ketogenic diet decreases epilepsy incidence.
You want to start?
Okay, fair enough.
I can also, I don't mind.
I love Paul's perspective that he is an optimist, because I think you have to be an optimist. I think we are a very, very long way from having the nutrient data that would allow us to do that. And I feel that the I guess my interest in nutrition and diet is what I would say is we are so far from eating a diet that is aligned with our best evidence about what a healthy diet should be in the UK, just in terms of fat, salt, sugar, and energy density and fiber and fruit and veg. We're so far away from that that the idea of drilling down and looking at the effect of individual molecules and encouraging their consumption on particular patients that feels sort of almost utopian at the moment. What we need from a cancer prevention point of view is to get people to start eating even 50% of the recommended amount of fruit and vegetables that they eat and halving their sugar consumption, and that would probably have the. So I'm looking at it's not that I'm a pessimist, but my optimism lies in trying to make these very sort of straightforward shifts in changing cancer biology. I must say I think we could work harder when we talked about the patient-reported outcomes. I do think we could work a lot harder on the diet of patients in hospital, and I think that probably would improve outcomes. But again, it's quite big picture changes from my point of view. It's not thinking about the interaction of little molecules with particular receptors. Simona, am I being too pessimistic?
No, I mean, I would say from a biological standpoint that we know that obviously diet has a major impact on inflammation within the brain. And as in many cancers, as we said before, inflammation is a key part of the problem. It's what promotes the tumor growth and facilitates the tumor formation in the first place and continues to support it. So anything that would lower that basal level of inflammation would be beneficial. And we know also that inflammation goes up with aging, and the more we age, the more our brain becomes inflamed. And then if on top of that we add poor nutrition that would, elevate those levels of inflammation, we're not doing well. But equally, we have an example. This is in animal models, so obviously it's a long way away from patients, but we know that nutrition could have a very significant impact. So we published a study a couple of years ago in which we showed that a fish oil high diet in animal models was sufficient to significantly delay tumorigenesis. And we worked out why the mechanisms, that were sort of underlying that. And it was all about how the cells of origin for, the cells that begin to form the tumor respond to this high fish oil diet by changing their metabolism. And that keeps them in check and makes them behave more like normal cells. And therefore, it delays the formation of a tumor for quite a long time. So I think we definitely have data in the lab that this is super important, and the key is to reduce inflammation. And it should be possible to do it through diet.
Thank you. It's very interesting. I have some questions for Paul now from our audience. So, do you think in terms of genetic engineering, iPS cells are playing a role for treatments in cancer? Well, let's answer that first.
So I suppose I better explain what iPS cells are for people. So that means induced pluripotent stem cell. So if you look, every cell in your body, right, comes from one cell in the first place, and the sperm and the eggs. So everything comes from that. So as a stem cell, it has the ability to make every possible thing. And what an iPS cell is when you take an adult cell and you make it go back into being a stem cell, and then you can then try and use that stem cell to make whatever cell type you want. Like I want to make a heart. I might try and make heart cells at least, or I want to make immune cells. So people have looked at trying to make immune cells. And the reason that this would be nice to do is because you get like a big bank of them that you could use potentially to sort of make in a oner and have an everlasting supply. That's really hard. So making a T cell, you can make something which looks like a T cell, and then it doesn't really work that much like a T cell when you put it into people. I mentioned that part of my work is to try and make kind of these universal donor T cells, and that's really hard as well because your immune system's great at rejecting things that are foreign, and when you put foreign healthy cells in, it recognizes them as foreign as well. So I think we're a long way off them being a therapy which is kind of approved these kind of T cells we make from iPS cells, but there's definitely a whole load of promise in it for trying to make this kind of scale. And you're trying to make these not like elite therapies which you can only give to rich Western countries that make them that you can treat lots of people in lots of different countries with them. That might be part of the answer, but we're a little way off.
Thank you. And off the back of that, another question for you. You mentioned using T cells from donors. So how do you prevent rejection by the patient?
I mean, yeah, so that's part of the $1 million question that we do. So T cells, or all the cells in your body, right, are covered in lots of molecules which are very unique to you. So that's why, for example, you know, if you're going to bone marrow transplant register, you have to have lots and lots of people on a bone marrow transplant register because the chance of finding someone who's got similar molecules to you is quite low. And the more people you've got, and the more chance you are of finding somebody. So all these T cells, along with every other cell, have got all these molecules that they're expressing, which are just basically like big flags to the immune system to say, look, you're foreign, you don't belong here, I'm going to kill you. And you know, that's, I don't know if I can say that out loud, but that's kind of what happens. And then what we try and do is hide those molecules, so that the immune system, the T cells particularly, don't see these molecules. And then possibly put in some decoy molecules and other things which allow the T cells to survive in what's ultimately quite a hostile environment. So these are really difficult. Proving that they work is really difficult. But that's kind of like part of what we do in the lab. So doing lots of lab experiments and that, doing some animal models, and yeah, hopefully eventually also with a clinical trial. There's also a clinical trial we have running at the moment, which is based on something we did in the lab to make these off-the-shelf CAR T cells.
Thank you for that, Paul. And a question I think to be directed at Kate, to do with AI. So has AI truly started playing a role in the early diagnosis of cancer, or is it being used more in other stages such as treatment and radiotherapy? And if so, how?
Well, I mean, the whole hope of AI in healthcare as a whole is that it can be used through all of the different parts of the patient pathway. So there's lots of research looking at, you know, making pattern connections between histopathology and genomics so that we can try and, you know, potentially even replace genomics with histopathology for, you know, the benefit of low middle income countries. So that's one kind of role. There's other roles for the early detection, so where you're collecting data across, you know, multiple timescales to see the evolution of different signals, how they grow, how they shrink, and how they are related to changes in imaging. So you're linking up all of these multiple data sets. And I think that's the real challenge, because these data sets are very diverse in where they come from. The types of data aren't easily put together. You can't easily integrate them. And it's very laborious to try and label images to, you know, for saying, okay, this part of histopathology shows this type of signal. So you do need people to do that. And there are ways and techniques that we're working around now to try and circumnavigate that, you know, the labor intensivity of all of this. So you can use it for early diagnosis, but I think the current take up seems to be more in clinical decision making. And it looks like, you know, where it's going in the next decade or so is that AI will be assisting doctors to make these decisions. You know, a doctor can't analyze millions of pieces of data in seconds to make decisions. They have intuition and they have clinical experience, but AI can do that. And so together, that pairing could actually be quite powerful to guide these decisions.
Thank you for that. And our next question is for Simona. So, Professor Parrinello, you mentioned that you're trying to build and come up with models and techniques to detect cancer earlier than the late stage detection which occurs these days. So, how can this be possible without patients themselves coming for early checkups? Because symptoms with brain cancer for example, become evident later on and when the cancer is already at an advanced stage?
Yeah, it's a really good question. So it's really challenging in brain because exactly of these reasons, but there, you can imagine two ways of thinking about this. One would be to intervene earlier, even in the context of a few months, just by refining the way it's diagnosed. And there's a lot of effort going on around this, which can be also linked to, training and education within sort of like GPs, for example, to better spot potential signs of cancer earlier. And this is something that can be done much faster, right? Sort of like in a very short time frame, and there is activity in this area. In the longer term, when we're talking about what the future might hold, the only way this would work is, well, there are two ways. One is you would identify risk groups of populations that would benefit from periodic screening. Of course, you would need a tool to screen, and this is where, doing the basic science is really important to find those ways. But there are certain risk groups that we know are likely to be more susceptible to brain cancer. Exposure to radiation is very well known, but there are some genetic disorders that predispose to brain cancer. So those populations could be potentially screened more regularly. But my hope is in the longer term, we would be screened regularly for most cancer types once we are able to develop those assays that are low cost, and it's sort of part of your general checkup. If it's about, for example, a blood-based screening system, right, that you would do. But that we're a very long way away from that. But I think we start by understanding the disease, and that's where we will then identify ways to do it. Does that make sense? I hope so.
It does. Thank you. Thank you very much. Our next question is for Chris. Are there any foods that have been found to have shown a protective effect against cancer? And if so, do we understand the mechanism behind these?
Well, I'd love the reflections maybe of the other people on the call, but there are, we know that just as we understand that a diet of industrially processed food that is energy dense, high in saturated fat, salt, and sugar is detrimental to health and is likely, I think there's good evidence it does increase the risk of certain cancers. So conversely, a diet of whole and minimally processed foods, of green leafy vegetables, fresh fruit and veg that's more plant-based and has fish in it, and the fish oils that Simona mentioned, we know that's protective against lots of diseases and probably cancers as well. There are some mechanisms that I think we understand fairly well. Fiber, and it's probably not just any old fiber, but the fiber that we get from the food matrix of fruit, veg, pulses, legumes, and whole grains, nourishes the microbiome. The microbiome in turn produces short chain fatty acids that then regulate immune function around the gut and around the body, has cardiac and brain effects as well. So, but we don't understand great detail about the links between the microbiome and cancer, other than to say it probably is important and eating sources of fiber are good for you. And we think some of the food additives and plastics also do increase the risk of cancer. So the emulsifiers particularly, emulsifiers are molecules that mix aqueous solution and fatty product molecules. So they act as detergents. And in simple terms, they can scrub out the gut, altering the microbiome, thinning gastrointestinal mucus, and leading to bacterial translocation, which sets up cycles of inflammation, which, as several people have said, is linked to cancer. So I think we've got quite a few different mechanisms. And then we have the inflammation of the different fats and weight gain and cardiovascular disease and so on. So we do understand the dietary pattern. I think where the difficulty comes, or at least my unease comes from going, a particular kind of food, a particular berry or a type of oily fish prevents cancer. I think that's really hard because people don't eat foods, we eat dietary patterns. And so I think when we talk about populations, the thing of interest is always to look at the overall diet and what it contains. And that I think we have good evidence about.
Thank you for that. I have
Say,
another
uh,
question.
say one thing. Yes, I think it's really interesting actually because you have such a difficult job, right? When we do, like, I want to see do my CAR T cells kill these things. I just get some CAR T cells and I put that there and I see does it kill them. But your experiments aren't like that, right? You can give mice like or whatever. You can give them goji berries or whatever you want and see what happens, or you can over give them something which is a carcinogen, and you can say, okay, well, if you give them huge amounts of it, it causes or doesn't cause cancer. But it doesn't tell you, as you said, whether consumption of that in your diet is good or bad. And you just have to have these huge populations and do very clever statistics to try and work it out, right? And I guess that's a good and a bad thing, but
Well,
yeah.
I mean, it's sort of axiomatic. The better the experiment, and we do put people in metabolic chambers and lock people up in labs
Yeah.
and control their diets, and the better the integrity of the experiment, the less applicable it is to the real world of people eating complex diets and living complex
Yeah.
lives. So, yeah, well, that's very nice of you to acknowledge the challenges of nutritional research.
Acknowledge the difficulty of working in that area. Yeah.
Curing cancer, a famously easy thing to try and do.
Super easy. Yeah. So easy.
Thank you. Thank you both. Chris, I actually have another question for you, which is quite interesting. Do you think there could be technological developments in food processing that are able to reduce the carcinogenic potential of foods without compromising their texture and taste, their shelf life and convenience? Or would you say a possible solution is to educate the public about the risks of UPFs and avoid them in our diets?
I don't think those two things are mutually exclusive. I think the thing that people forget is we know very well how to invent convenient, cheap, healthy food. It can be done easily. You can go to shops and buy it. It's incredibly expensive. It's not marketed very heavily compared to the addictive, very cheap food. So the problem with food is not the theoretical design. We know what a healthy diet looks like. The problem is the foods that survive in the marketplace are the ones that are very cheap to make and have addictive properties. And as long as the market depends on excessive and increasing consumption, which is what financialization means, it's very hard to imagine how you design a successful food product that survives in the marketplace that doesn't lead to excess consumption. I mean, there's a lot more economics and mathematics around that, but it's sort of like going, could we invent a healthy cigarette? I mean, yeah, you can take out the nicotine, you can take out the tobacco, you can do all kinds of things, but if the cigarette isn't addictive, it's not going to survive in the marketplace.
Thank you. And just on that note, we're running out of time, so we're going to have to conclude here. So I just want to say a massive thank you to everyone for joining us. So thank you to all of our panelists. It was fascinating hearing about your respective fields, and I think this discussion was incredibly interesting. And with the fast rate of development of technologies and research, it seems like it's a very exciting time to be a scientist right now in this particular field. And to our audience members, thank you for joining us and taking part in this discussion and giving us your time this evening to do so. And before you go, we kindly request your feedback on today's event if possible. So a survey will be sent to you shortly after this event, and we greatly appreciate your input. Also, if today's event has inspired you to explore graduate studies in the field of cancer, if you're currently an undergraduate, we warmly invite you to our virtual Cancer MSc Open event on Wednesday next week. So you can find the link to this event in our chat. So hopefully that'll be shared soon. And on that note, I would just like to once again say thank you to everyone for coming and taking part. And it's been a really interesting hour of discussions. And I wish you all a wonderful evening.